This is Heliox, where evidence meets empathy. Independent, moderated, timely, deep, gentle, clinical, global, and community conversations about things that matter. Breathe easy, we go deep and lightly surface the big ideas.
I want you to close your eyes for a second, assuming you're not driving, of course, and just imagine a scenario for me.
Okay, assuming we're safely parked.
Right, safely parked. So you are going about your normal Tuesday. The weather is fine, the commute is totally routine, and then suddenly there's this jarring, violent interruption.
Like a car crash.
Yeah, maybe it's a minor fender bender on a rain-slicked road where your head just whips forward and back. Or maybe you're playing a weekend pickup game of soccer. You go up for a header and you collide with another player.
Or, honestly, even something as simple as slipping on a patch of ice in your driveway.
Exactly. And the initial impact in all those scenarios...
We were talking about milliseconds. Just a literal fraction of a second.
Right, a fraction of a second, and then it is over. You sit up, you rub your head, you might have a dull ache, maybe a little bruise forming near your hairline, but you shake it off.
Because you feel lucky, you know?
Yeah, you survive the impact. But what we are going to explore today in this deep dive is the terrifying reality that the initial impact is, well, it's not necessarily the main event.
Not even close in a lot of cases.
Right. What if the most devastating damage from that collision isn't the physical bruise on your forehead? What if that really brief mechanical force ignited like a slow-burning biological fire deep inside your brain tissue?
A fire that doesn't really reach its destructive peak until months or, even years after you have completely forgotten about the ice patch.
Yeah, that is exactly what we're looking at today. Because framing it as a slow-burning fire, it really shifts the whole paradigm of how medicine has historically viewed physical trauma, doesn't it?
It completely flips it on its head. For generations, the clinical assumption was quite linear, right? An injury occurs, a wound is created, you stabilize the patient, and from that moment onward, the body is basically in a state of healing.
Like the damage is done and over with.
Exactly. The damage is considered a static artifact of the past, but traumatic brain injuries, or TBIs, they just totally defy that logic. That initial fraction of a second is literally just the ignition source.
The spark.
Yeah, the spark. The resulting damage is this actively evolving landscape. It's an ongoing event.
And we really need to grasp the sheer scale of this before we get into the microscopic details.
Okay.
Because the numbers we are looking at in the source material, they are pretty sobering.
They're massive.
Yeah, globally, researchers estimate that 69 million people suffer a traumatic brain injury every single year.
69 million, that's a staggering figure.
Just picture that,
Yeah.
That is roughly the entire population of the UK or France experiencing a brain injury every 12 months. And in the US alone, the CDC tracks about 2.8 million injuries annually.
Which translates to over 200,000 hospitalizations and more than 50,000 deaths.
Right. And an annual economic burden of something like $76.5 billion.
Yeah, that economic figure is just massive. It accounts for emergency care, long-term rehabilitation, lost productivity, all of it.
But the demographic breakdown is what I found truly interesting. It illustrates the universality of the risk. Because I think people assume TBI is just a sports thing.
Right, they think of high-contact athletes or military personnel. But the highest incidence rates actually bookend the human lifespan.
Toddlers and the elderly, right?
Exactly. You see massive spikes in TBI among those two groups, primarily driven by accidental falls, just tripping and hitting your head.
And then sitting right in the middle of that distribution, you have teenagers and young adults.
Where the injuries are predominantly motor vehicle crashes and sports-related incidents, yeah.
And as massive as those figures are, the sources say they are likely a severe undercount.
Oh, without a doubt. Think about it. If you slim and bump your head but don't lose consciousness, you probably don't go to the ER.
Yeah, you just take some ibuprofen and go to sleep.
Right. So mild traumatic brain injuries, which are commonly known as concussions, they often go entirely untreated and unrecorded.
Which means we are looking at this colossal public health footprint, this vast shadow population of individuals who are managing long-term, sometimes lifelong, cognitive and emotional recovery.
Which leads us directly to the core mystery of the whole thing.
Why do people who suffer a seemingly mild brain injury go on to develop chronic depression, severe anxiety, memory loss, and, cognitive decline long after the physical tissue should have healed.
Right, why is the brain still sick years later?
That mystery is the focal point of today's deep dive. We are exploring a really fascinating, comprehensive scientific review paper. It's titled Deplete and Repeat.
A great title, honestly.
It is. And the researchers behind this paper are looking at a radical, almost sci-fi approach to treating TBI. They pose this highly controversial question.
Basically asking, what if the brain's own highly specialized immune system is the very thing preventing the brain from healing?
Yeah. What if the microscopic security guards that are literally meant to protect your neurons are actually the culprits slowly destroying them?
It's a huge betrayal by your own biology.
And most incredibly, what happens when scientists use a highly targeted drug to completely obliterate that immune system, essentially forcing the brain to grow a brand new one from scratch?
It totally challenges fundamental assumptions about immunity and neurology.
So we're going to journey into the microscopic landscape of your brain today. We're going to meet these shape-shifting immune cells, watch them become dangerously paranoid, and then examine the mechanics of a drug intervention that sounds, honestly, like a controlled biological demolition.
But to understand how to fix the brain, we first need to really understand the physics and the biology of how it breaks.
Right, so let's start there. How does it break? The breaking process is bifurcated, right?
Yeah, a traumatic brain injury is officially classified into two distinct cascading phases. The first phase is what we call the primary injury.
Okay, the primary injury.
This is the direct, instantaneous result of the mechanical force applied to the skull.
Let's visualize that physical force for you, the listener, because your brain is not just rigidly bolted to the inside of your skull.
Right, it's not screwed in.
It is essentially a gelatinous, highly vascularized mass. And it's floating in this cushion of cerebrospinal fluid encased within a hard, unforgiving, bony vault.
And that physical arrangement is actually great for everyday shock absorption. Like when you are jogging, your brain is just gently bobbing around in that fluid.
But it becomes a huge vulnerability during extreme, rapid acceleration or deceleration.
Exactly. When you hit your head or when your body comes to a sudden halt in a car crash, your skull stops moving instantly. But your brain continues its forward momentum.
Physics just takes over.
Yeah, it crashes into the front of the skull, and then it rebounds and crashes into the back of the skull. We actually call this a coup contre coup injury.
And it isn't just forward and backward motion, is it? Because the source material specifically highlights rotational forces as being really bad.
Oh, rotational forces are often the most destructive. When the head twists violently, the brain rotates within the skull. And the issue is that the brain isn't perfectly uniform in its density.
Right, you've got different types of tissue in there.
Exactly. The outer layer, the gray matter, has a completely different density than the inner wiring, the white matter. So when rotational force is applied, these different densities actually move at different speeds.
That sounds like it would cause things to tear.
It creates massive shear strain. Imagine holding a bundle of raw spaghetti in your hands, right? And you violently twist the two ends in opposite directions.
Oh, the strands in the middle are just going to snap immediately.
Precisely. In the brain, those strands are axons. They are the long, really delicate cables that neurons use to transmit electrical signals to one another.
So the mechanical shear force just twists, tears, and physically severs these communication cables.
It does. And the cell membrane, which is normally this tightly sealed protective barrier, is just violently ripped open.
Wow, so all the internal contents of the cell just spill out into the surrounding tissue.
Yeah, it's a microscopic spill. And at that level, that immediate tearing is totally catastrophic. Crucial ion channels are destroyed, which causes this massive, unregulated influx of intracellular ions, things like calcium.
Which is bad, I assume.
Very bad. The cell death in this primary phase happens within minutes. And honestly, with our current medical technology, this initial physical destruction is irreversible. You just cannot untear an axon.
So I am picturing a massive earthquake hitting a major city. The primary injury is the tectonic shift itself. The ground shakes violently and the buildings instantly collapse.
That's a really good way to think about it.
Because there is nothing you can do to stop the earthquake once the fault line slips, right? And there's no way to instantly rebuild those collapsed structures. The destruction is just this harsh, immediate physical reality.
The earthquake metaphor is incredibly apt because of what happens next. The earthquake ends, but the disaster is far from over. This is what initiates the second phase, the secondary injury.
The secondary injury is the aftermath. So the earthquake ruptured the city's infrastructure. Now, you have broken gas lines sparking raging fires. You have ruptured water mains flooding the streets.
The electrical grid is short-circuiting.
Exactly. The emergency response system gets completely overwhelmed, leading to chaotic, uncontrolled damage that spreads way, way beyond the footprint of the originally collapsed buildings.
And in a real city, the fires and the flooding can often destroy far more property than the initial tremor did.
Right. So how does that translate to the brain?
In the brain, the secondary injury is this chronic, progressive, and highly toxic biochemical response to the initial mechanical trauma. The sources detail the specific biological fires that break out.
Okay, let's talk about those fires.
Remember the torn cells we just discussed? When a neuron is ripped apart, it doesn't just die quietly. It violently ejects its cytoplasmic and nuclear proteins into the surrounding extracellular space.
And the brain is a highly regulated environment, right?
Yeah.
So imagine random internal proteins floating around sets off major alarm bells.
Oh, absolutely. These spilled proteins act as what we call DAMPs. That stands for damage-associated molecular patterns.
DAMPs. Okay, so what do?
Think of DAMPs as chemical distress flares. They signal to the entire surrounding biological neighborhood that a catastrophic breach has just occurred.
So the distress flares go up. What else is spilling out of these ruptured cells besides proteins?
Neurotransmitters. Specifically, a massive continuous release of excitatory neurotransmitters, mostly glutamate.
Now, glutamate is normally a good thing, right?
Yes, glutamate is absolutely vital for normal brain function. It's quite literally how neurons signal each other to fire. But in high, unregulated concentrations outside the cell, it becomes a potent toxin.
It's like someone screaming, "Fire!" in a crowded theater. A little communication is good, but widespread panic just causes a deadly stampede.
That phenomenon is literally called excitotoxicity in neurology.
Excitotoxicity, that is a great word.
It is. The surviving neurons in the area receive so much glutamate signaling that they just start firing uncontrollably. They become hyperactive to the point of utter exhaustion and death.
Wow. So they basically work themselves to death because of the panic.
Exactly. Which obviously spreads the zone of cell death outward from the initial impact site. Furthermore, you start to see severe mitochondrial dysfunction.
Okay, mitochondria. Bringing back high school biology here. Those are the power plants of the cell, right? Generating the ATP, the energy currency, needed for survival.
You got it. When excitotoxicity hits, the massive influx of calcium into the surviving cells just completely overloads the mitochondria. The power plants fail.
So the lights go out?
The lights go out. The cells cannot produce energy, leading to further necrosis. And on a macrostructural level, the blood-brain barrier begins to physically break down and leak.
Wait, the blood-brain barrier. That's the tightly woven network of endothelial cells that normally acts as a strict security checkpoint, right?
Yeah.
Keeping the body's peripheral immune system out of the brain.
That's the one. It starts to crumble.
So the city is on fire, the power grid is down, the citizens are panicking to death, and the security walls protecting the city have basically fallen over.
It is total biological chaos.
And the review paper emphasizes that this secondary phase isn't just like a brief chemical storm that blows over in a few hours.
No, the timeline is actually the most critical factor here. These secondary sequelae, the simmering biochemical fallout, it can persist for months, and in many cases, years.
Years.
Yes, it is an ongoing, evolving, degenerative process. This persistent damage is what creates that massive population of survivors we mentioned earlier who are dealing with chronic issues.
Right, so the chronic neuroinflammation is the actual biological engine that is driving the long-term cognitive deficits, the social impairments, and the development of severe psychiatric disorders years after the initial physical impact.
That is exactly what the data suggests.
But, the fact that it's an ongoing process, that actually offers a glimmer of hope, right?
How so?
Well, if the primary injury is an unstoppable earthquake, but the secondary injury is a slow-spreading fire, fires can be fought, fires can be extinguished.
I see what you mean, yes.
Because the secondary damage takes months to unfold, it creates this massive therapeutic window. If we can just figure out what is fueling the fire, we can intervene and actually alter the long-term prognosis for the patient.
That is the exact goal of this research. But finding the source of the fire requires us to identify the firefighters first.
Right. Who is responding to these damps, these biological distress flares? Who is supposed to be managing this crisis?
This introduces the absolute star of today's deep dive, a cell that is basically both the hero and the ultimate villain of the TBI story, the microglia.
The microglia. I honestly had never heard of them before reading the source material, but they are wild.
They are among the most fascinating and complex cells in the entire human body. They make up roughly 10% of the total cell population in a mature central nervous system.
10% is a lot.
It is. Functionally, they are the resident innate immune cells of the brain and the spinal cord. They are basically the brain's dedicated security force and waste management team rolled into one.
And I found their origin story in the literature to be completely mind-bending because they are brain cells, obviously, but they don't actually originate with the rest of the brain, do they?
They do not, and this is so cool. Most cells in the central nervous system, your neurons, your astrocytes, oligodendrocytes, they all develop from the neuroectoderm. That's the embryonic tissue that folds to become the neural tube.
Right, the standard brain starter kit.
Exactly. But microglia have a completely separate lineage. They are derived from early progenitor cells located in the embryonic yolk sac.
The yolk sac. Like, wait, so they develop outside the embryo entirely in the structure that provides early nourishment, and then they have to physically travel into the developing brain.
Yes. Very early in embryonic development, before the blood-brain barrier is fully formed and sealed off, these yolk sac progenitors literally migrate into the developing central nervous system.
That is incredible.
And once they enter, the gates just close behind them. The blood-brain barrier matures, it seals shut, and these migrating cells differentiate into microglia.
So they basically sneak in while the walls are still being built, and then they're locked inside.
That's exactly it. And once they're in, they are remarkably self-sustaining.
How so? Like, don't they die and get replaced?
Well, unlike peripheral immune cells in your blood, which are constantly being regenerated by your bone marrow every day, microglia are incredibly long-lasting.
Oh, I see.
They have an extremely low turnover rate in both rodents and humans. They rely entirely on self-renewal through localized cell division.
So it's a closed system.
Exactly. The population that seeds your brain during fetal development is essentially the ancestral population that maintains your brain's immunity for your entire life.
Wow. Okay, so let's examine how they function when everything is going well. We need to find the baseline for you guys listening. In a perfectly healthy brain, prior to any injury, what do these microglia look like, and what are they doing?
Under homeostatic, healthy conditions, microglia exist in a state that neurobiologists call ramified.
Ramified, meaning branched out.
Exactly. Visually, a ramified microglial cell has a very small, stationary cell body, but it extends numerous, long, incredibly delicate, highly complex branching arms.
Or like a tree.
Like a very active tree. Because these arms are not static, they are in constant rapid motion, perpetually extending, retracting, and sweeping through the brain's parenchyma.
Let's define parenchyma for our listeners quickly.
Yeah.
The parenchyma is essentially the functional meat of the brain tissue, right? The dense forest of neurons and synapses, as opposed to the structural scaffolding or the blood vessels.
Right, and the microglia use these long, sweeping branches to constantly monitor that functional tissue. They are the ultimate neighborhood watch.
Just patrolling the streets.
Yes. They survey the environment, scanning for any microscopic signs of damage, metabolic waste, or invading pathogens. They even interact delicately with synapses, the connections between neurons, occasionally pruning away weak connections to optimize brain function.
I picture them like a microscopic, highly sensitive octopus.
An octopus is a great visual.
Right. The main body just sits quietly in a crevice, but its long tentacles are constantly, gently feeling the surrounding water, checking the neighborhood.
That is exactly what they do.
But then the earthquake happens. The primary mechanical force of a TBI shears the axons. The cells rupture. The damps, those distress flares we talked about, they flood the surrounding fluid. The tentacles of our microscopic octopus feel those flares. What happens next?
The transformation is immediate, and it is dramatic. When the ramified microglia detect these danger signals, they undergo rapid phenotypic and transcriptional changes.
They change their shape.
They completely abandon their delicate sweeping form. They retract those long branching arms right back into the center, their cell bodies swell and enlarge, and they transition into what is termed an amoeboid or hypertrophic state.
So they go from an elegant branching octopus to a bulky, aggressive blob.
A highly functional blob, though. In this amoeboid state, their primary objective is containment and clearance. They physically migrate to the exact site of the injury.
They rush to the scene.
Right. And once there, they begin clearing out the cellular wreckage, the dead neurons, the spilled myelin, the toxic debris. They do this through a process called phagocytosis.
Phagocytosis, which translates roughly to cell eating.
They literally engulf the dead tissue, draw it into internal compartments called lysosomes, and dissolve it using highly acidic enzymes.
Like little Pac-Man.
Basically, they are acting as a microscopic cleanup crew, rapidly removing the toxic debris to prevent it from damaging the surrounding healthy neurons.
But they aren't just eating, though, are they? The paper says they are also actively coordinating the biological response team.
Yes, while they are phagocytosing the debris, they are also synthesizing and releasing pro-inflammatory cytokines and chemokines.
More chemicals.
Right, these are specialized signaling proteins. They essentially recruit other local glial cells, like astrocytes, to the area to help wall off the damage and begin the tissue repair process.
Okay, I have to pause here and play devil's advocate for a second.
Go for it.
We just established that the microglia sense the danger. They transform into a robust cleanup crew, they eat the toxic debris that would otherwise poison the brain, and they coordinate the repair process.
Yes.
This sounds like an incredibly efficient, highly necessary, and fiercely neuroprotective biological response. So why does the review paper suggest we need to stop them? If I'm a patient bleeding in the street, I don't want someone to come along and fire the paramedics while they are actively pulling people from the rubble.
Your logic is perfectly sound, and it's exactly why this field of research was stalled for decades.
Really? Because researchers assumed they were only helping.
Yes. The acute inflammatory response, the paramedics pulling people from the rubble, is absolutely neuroprotective and necessary for survival. You need them there on day one.
Okay, so what's the problem?
The paradox and the really insidious twist in the biology of traumatic brain injuries is that the microglia do not know how to stand down once the acute crisis is resolved.
What do you mean they don't stand down? The debris gets cleared eventually, right?
The physical debris gets cleared, yes. But the cellular trauma fundamentally alters the microglia. TBI causes these cells to enter a state known as microglial priming.
Priming, like priming a pump to draw water, or like priming an explosive to detonate.
The explosive analogy is much closer to the biological reality here. After the initial injury phase passes, the surviving microglia do not revert back to their peaceful, delicate, ramified state.
They don't go back to being an octopus.
No, they get trapped in this intermediate, hypervigilant state. They stop producing the massive wave of inflammatory cytokines, so the acute fire seems to die down on the surface, but their internal transcriptional programming remains permanently altered.
Can we break down what that altered programming actually looks like? The source mentions they upregulate certain markers, specifically CD68 and MHCII. What do those acronyms actually mean in terms of the cell's behavior?
Sure, let's start with CD68. This is a protein heavily expressed on the membrane of lysosomes, which, as we discussed, are the cell's digestive stomachs. So a high presence of CD68 indicates that the cell is drastically ramping up its digestive machinery. It is hungry, and it is preparing to consume large amounts of material.
So they're walking around months later with a hyperactive digestive system, just looking for something to eat. And what about MHCII?
MHCII stands for Major Histocompatibility Complex Class II. I mean, think of it as a biological wanted poster.
A wanted poster.
Yeah. Cells use MHCII to present fragments of pathogens to other immune cells, essentially signaling an active threat. When microglia heavily upregulate MHCII in the absence of a real infection, it means their threat detection threshold is practically zero.
To synthesize this for the listener, the TBI forces the microglia into a state where they are biochemically starving, armed to the teeth, and holding up wanted posters for enemies that don't even exist.
That is a perfect summary. They are utterly paranoid.
Wow.
And here is where the tragedy of the secondary injury truly unfolds. In the life of a human being surviving a TBI, secondary stressors are just unavoidable. We refer to these as secondary immune challenges.
What qualifies as a secondary immune challenge?
Yeah.
We aren't just talking about getting hit in the head a second time.
No, not at all. These challenges can be remarkably mundane. For instance, a severe disruption in circadian rhythms and sleep architecture.
Which is a really common symptom following a concussion, right?
Very common. And lack of sleep acts as a systemic stressor. Or experiencing intense chronic psychological stress, perhaps from the financial burden of the injury or social isolation. That causes the release of stress hormones that cross the blood-brain barrier.
Right. What else?
It could be a simple peripheral infection, like contracting the seasonal flu, catching a cold, or just getting a stomach bug.
So the person has a TBI, months go by, their brain feels mostly fine, but their microglia are secretly sitting there, paranoid and heavily armed. Then, 6 months later, the person catches a bad cold or pulls 3 all-nighters in a row.
Right.
What does that secondary stressor do to the primed microglia?
It acts as the trigger on the explosive. Because the microglia are primed, they do not mount a proportional normal immune response to the cold or the lack of sleep. They overreact massively.
They freak out.
Totally. This mild stressor provokes an exaggerated, disproportionate release of highly toxic inflammatory molecules.
What kind of molecules are they unleashing?
Well, they release a flood of pro-inflammatory cytokines, but more destructively, they release reactive oxygen species and nitric oxide.
Which sounds bad.
Very bad. These are highly volatile cytotoxic compounds that cause severe oxidative stress. They literally shred the lipid membranes and DNA of surrounding cells.
But wait, earlier you said they were eating dead debris. Now they were just spraying toxic chemicals everywhere. What happens to the healthy brain tissue caught in the crossfire?
It is decimated by friendly fire, and it gets worse.
How could it get worse?
Remember how CD68 indicated they were hungry? In this hyperreactive state, their phagocytic drive, their urge to eat goes into overdrive, but they lose their target specificity?
Meaning they stop caring what they eat.
Exactly. They stop looking for dead debris and start engulfing and digesting perfectly healthy tissue. Specifically, they target and consume healthy synapses, the vital communication bridges between living neurons.
Oh my God.
Yeah.
They're literally eating the victim's memories and cognitive function.
Yes.
The firefighters haven't just become arsonists, they have become a hyper-aggressive demolition crew tearing down intact homes because they were convinced a fire might start tomorrow.
It's tragic. And this widespread synaptic loss and chronic neuroinflammation, it directly correlates with the onset of delayed cognitive deficits, memory impairment, and severe mood disorders. It is a terrifying biological mechanism.
It is terrifying. But the source material introduces a really fascinating, deeply important nuance to this microglial behavior that we absolutely have to discuss.
The sex differences.
Yes. The paranoia, the friendly fire, the resulting depression, it doesn't manifest identically in everyone. There's a profound sex difference in how microglia operate.
This is one of the most rapidly evolving and crucial areas in modern neuroimmunology right now. For decades, neuroscience largely ignored biological sex as a variable, mostly just studying male rodents.
Which is a whole other issue we could do an entire deep dive on.
Absolutely. But we now know that adult male and female microglia possess fundamentally different baseline transcriptional profiles. They are, at a genetic level, programmed differently.
How does that programming differ before an injury even occurs? Like just walking around on a normal Tuesday?
It actually traces back to their developmental role. Microglia are critical mediators of sexual differentiation in the developing embryonic brain.
Oh, really?
Yeah. And because of this, even in healthy adult brains, female microglia naturally express higher baseline levels of genes associated with cellular repair, development, and cytoskeleton organization.
And the males?
Male microglia, conversely, maintain a higher baseline expression of genes related to inflammatory pathways and immune activation.
So the male cells are naturally walking around with their hands a little closer to their holsters, basically.
That's a fair way to put it.
So how does this subtle baseline difference play out when the catastrophe of a TBI actually hits?
We see a distinct divergence in the timeline and nature of the pathology. In human clinical data, males generally have a higher overall incidence of TBI and a higher acute mortality rate.
So they die more often initially.
Yes. However, females who survive the injury consistently report higher severity and longer duration of post-concussive symptoms, particularly regarding depression, anxiety, and persistent physical symptoms like chronic headaches.
And researchers can trace this specific difference back to the microglia. Like it's not just hormones.
They can model it incredibly well in preclinical rodent studies to prove it's the microglia. When scientists subject male and female mice to identical traumatic brain injuries, the initial immune response looks totally different.
Okay, walk me through it. What do the male mice do?
Male mice typically show significantly more acute early neuronal cell death, intense astrogliosis, and a massive rapid spike in microglial activation within the first few days.
So an immediate violent cellular fire. And the females?
Female mice often do not exhibit that massively exacerbated early physical cell death. It's much calmer initially, but the pathology is simply delayed and it's shifted towards psychiatric outcomes. When researchers evaluate the mice weeks later, say, a month post-injury, the female mice display significantly more delayed cognitive impairments and profound depressive-like behaviors compared to the males.
Wait, how do researchers even measure depression in a mouse? I mean, you can't hand a mouse a psychological questionnaire.
Obviously not. Researchers use highly validated behavioral assays. One primary method is the tail suspension test.
The tail suspension test, that sounds awful.
It's brief, but it's very informative. If you suspend a mouse by its tail, a healthy, resilient mouse will continuously struggle trying to climb up and escape the situation.
Which makes sense.
Right. But a mouse exhibiting a depressive-like state, suffering from a lack of motivation and behavioral despair, will basically give up and simply hang immobile much faster and for much longer periods.
That is heartbreaking to visualize.
It is. But female mice post-TBI show dramatically increased immobility times compared to males.
Which clearly demonstrates the delayed psychiatric toll. But again, how can we be absolutely sure the microglia are driving this sex-specific difference and not, hormones floating around in the blood like estrogen or testosterone?
Because of some truly elegant cross-transplantation studies highlighted in the review.
Oh, this part was wild.
Yeah.
Explain what they did.
Researchers literally took female microglia and surgically transplanted them into male brains.
Just swap them out.
Yep. And they found that these female microglia did not conform to their new male environment. They maintained their inherent female-specific gene expression profile.
So they didn't care that they were bathing in testosterone now.
Not at all. In models of ischemic stroke, possessing female microglia actually proved to be neuroprotective for the male brains, altering the injury outcome.
They carry their biological identity with them regardless of the host environment.
Exactly. It proves that the biological sex of these immune cells intrinsically dictates how the brain will mount an inflammatory response and recover from physical trauma.
Which means any future pharmacological treatment that attempts to modulate microglia simply must account for sex as a foundational biological variable.
Or it risks being completely ineffective or even harmful to half the population.
Right. Okay, let's anchor ourselves in the narrative here for the listener. We have established that the primary mechanical damage of a TBI is brief. The real danger is the secondary injury driven by microglia.
Correct.
These cells start as heroes, clearing debris, but the trauma mutates them. They become primed, hypervigilant, and paranoid. When a mild stressor comes along months later, they overreact, spewing toxic chemicals and physically eating the brain's healthy synaptic connections, leading to chronic cognitive and emotional decline.
That's the timeline, yes.
So the multi-billion-dollar question for the scientific community becomes obvious. How do we stop them? How do you disarm a paranoid embedded immune system that is literally holding the brain hostage?
Well, the initial impulse from the scientific community was basically just blunt force. If the microglia are the problem, find a way to kill them.
Right.
This approach is called pharmacological depletion. But the early tools researchers had were incredibly clunky and deeply flawed.
What did the early attempts look like? The review paper mentioned something called claudronate liposomes. And I have to say, liposomes sound like an expensive anti-aging cream.
They do sound like that, but their function here was lethal. A liposome is essentially a microscopic, artificially constructed bubble made of lipids or fats.
Okay, a fat bubble.
It is hollow on the inside, allowing scientists to package drugs within it. Claudronate is a type of bisphosphonate, which is a class of drugs normally used to treat bone density disorders like osteoporosis.
Why put a bone drug inside a fat bubble and put it in a brain? That makes no intuitive sense.
Because claudronate, when highly concentrated inside a cell, disrupts the production of ATP. It essentially suffocates the cell from the inside out and forces it to undergo apoptosis, or programmed cell death.
Oh, I see.
The liposome acts as a Trojan horse. Remember, microglia love to phagocytose, or eat fatty cellular debris.
Right. So scientists just introduced these fat bubbles filled with poison. The microglia eat the bubbles dissolve inside them, the claudronate is released, and the microglia die.
That's the mechanism.
That sounds like an incredibly clever microscopic assassination. Why did it fail?
It failed spectacularly for several structural reasons. First, claudronate liposomes are physically too large to cross the blood-brain barrier.
So you can't just swallow a pill?
No, you cannot inject them into the bloodstream either. Scientists had to physically drill into the skull and inject these liposomes directly into the brain's ventricles or parenchyma.
Which is inherently causing another traumatic brain injury just to deliver the drug.
Exactly. You are causing physical trauma to treat trauma. Second, it lacked cellular specificity.
Meaning it killed other stuff.
Right. Microglia aren't the only cells that eat things. Any cell capable of phagocytosis, including peripheral macrophages that might be nearby, would eat the liposomes and die.
That's messy.
But the fatal flaw was the biological reaction. The sheer mechanical trauma of injecting the liposomes, combined with the messy death of the cells, severely damaged the blood-brain barrier.
Making things worse.
Much worse. Instead of calming the brain down, the claudronate liposomes actually triggered a massive uncontrolled spike in pro-inflammatory cytokines.
The intervention meant to put out the fire acted like a chemical accelerant.
Precisely. They tried other blunt instruments as well, like a compound called MAC1 saporin.
Saporin. That sounds like a plant extract.
Good catch, it is. It is a highly potent ribosome inactivating toxin derived from the soapwort plant. It basically halts protein synthesis, killing the cell.
Now, what's the MAC1 part?
MAC1 is a receptor protein found on the surface of myeloid cells, including microglia.
Yeah.
The idea was to attach the plant poison to a molecular key that only fits the MAC1 lock.
Oh, okay, so a microscopic smart bomb tracking the specific target.
In theory, yes. In practice, it suffered the exact same limitations. It required invasive intracerebral injections. The depletion was localized and incredibly transient. It still wasn't perfectly specific to microglia, and once again, the administration itself caused severe blood-brain barrier disruption and further neuroinflammation.
So the field was basically at a dead end. They realized they couldn't just drop poison onto the brain. They needed a scalpel, but all they had in their toolkit were sledgehammers.
That's exactly right. And then the breakthrough occurs.
Yes. The review paper pivots to what we call the Control-Alt-Delete drug, the elegant scalpel. How did researchers finally figure out how to selectively turn off a microglia without tearing the brain apart?
The paradigm shifted when researchers stopped trying to poison the cells from the outside and instead looked at what the cells fundamentally require to survive from the inside.
Okay.
They mapped the precise biological signaling pathways of the microglia and zeroed in on a specific receptor located on the cell's surface, the colony-stimulating factor 1 receptor, or CSF1R.
CSF1R. What makes this specific receptor so crucial?
CSF1R is a transmembrane tyrosine kinase receptor.
Okay. Give that to me in non-jargon terms, please.
Fair enough. It is essentially an antenna that receives specific survival signals from the environment and transmits them into the cell's nucleus. For microglia, signaling through this specific antenna is absolutely non-negotiable.
What happens if it stops?
Well, it regulates their proliferation, their differentiation, and most critically, their baseline survival.
So it is literally their biological life support cord.
Yes. If you block that receptor, if you jam the signal to that antenna, the microglia cannot sustain themselves. They undergo rapid, silent, programmed cell death without triggering a massive inflammatory alarm.
So researchers just needed to find a chemical that clogs up that specific antenna.
And pharmaceutical science delivered. They developed highly specific small molecule inhibitors designed to target and block the CSF1R kinase domain.
Did it work right away?
Early iterations had some off-target effects, meaning they accidentally blocked other similar antennas, but the chemistry was rapidly refined over a few years. The current gold standard in the field, widely used in these preclinical TBI models, is a drug called PLX5622.
So PLX5622 is our sniper rifle.
Yeah.
How efficient is it? Let's say I am a researcher and I want to wipe out the microglia in a mouse model. Do I still have to drill a hole in its skull?
That is the most revolutionary aspect of PLX6232. It is a highly lipophilic small molecule, meaning it dissolves easily in fats, and it is incredibly tiny.
Okay.
Therefore, it crosses the blood-brain barrier with remarkable efficiency. You do not need needles. You do not need surgery. Researchers literally just mix the PLX5622 powder into the rodent's daily food chow.
Wait, they just eat it.
They literally eat their normal diet entirely unbothered.
And it works.
And the pharmacokinetic results are staggering. At the standard research concentration, if a mouse simply eats this medicated chow for 3 consecutive days, it completely and silently depletes 80 to 90% of the entire microglial population across the entire brain and spinal cord.
Okay, I am going to push back hard here,
Yeah.
because this is the point where the science sounds like absolute madness.
Go ahead.
We have established that microglia are the brain's dedicated immune system. They are the security force. You were telling me that by feeding a mouse a specific kibble for a long weekend, researchers are completely dissolving 90% of the brain's entire defense network.
That is exactly what they're doing.
If I have a human patient in an intensive care unit recovering from a severe car crash, my body is surrounded by opportunistic hospital pathogens. Bacteria are everywhere.
True.
If you give me a pill that wipes out 90% of my brain's immune cells overnight, aren't you leaving the central nervous system completely undefended? When a minor bacterial infection easily cross the blood-brain barrier and cause a fatal case of meningitis within hours?
It is a brilliant and totally necessary question, and it highlights the profound physiological tightrope this whole intervention walks. You are entirely correct to worry about the neighborhood when the police force vanishes.
Because the brain is not a static vacuum.
Exactly. The sudden mass disappearance of 10% of the total cellular population absolutely triggers compensatory shockwaves across the entire nervous and immune system.
So how does the local neighborhood, the other brain cells, react to the sudden absence of their security force? Let's look at astrocytes, for example.
Astrocytes are a phenomenal example. They are these large, star-shaped glial cells that perform immense structural and metabolic support in the brain.
What exactly do they do?
They wrap their tendrils around blood vessels to help maintain the physical integrity of the blood-brain barrier, and they regulate the flow of nutrients to neurons.
And how do they normally interact with the microglia?
They're essentially partners in inflammation. During a TBI, when microglia become hyperreactive and release those cytokines, the astrocytes detect those cytokines and also become reactive, entering a state called astrogliosis.
Yeah.
They swell up and form a dense fibrous scar, known as a glial scar, to physically wall off the injured tissue. They egg each other on, basically.
So if we use PLX5622 to suddenly assassinate all the microglia, what do the star cells do? Do they relax or do they panic?
Well, the timeline dictates their reaction. Acutely, in the first few days following the depletion, the astrocytes actually calm down.
Really?
Yeah, because the inflammatory screaming from the microglia is silenced, the early reactivity of the astrocytes is significantly reduced.
Well, that seems like a positive outcome. Less scarring, less immediate swelling.
It is beneficial initially. However, biology abhors a vacuum. If you observe the brain chronically, say, 30 to 60 days post-depletion, the astrocytes begin to alter their behavior.
They notice the cops are gone.
Yes. They recognize that the cellular debris is not being cleared. They sense the absence of the microglial baseline maintenance. Consequently, the astrocytes become hyperactive.
What?
They drastically upregulate their own gene expression, attempting to enact compensatory mechanisms. They stretch themselves thin, trying to act as makeshift microglia to help the injured tissue recover.
Which probably doesn't work out perfectly.
No, it can lead to its own set of long-term structural complications.
It's like the civilian neighborhood watch trying to do the job of a highly trained SWAT team. It's noble, but probably inefficient.
That's a great analogy.
And what about the body's peripheral immune system? Does the rest of the body realize the brain's gates are unguarded?
The peripheral immune system is acutely aware. There is constant, highly regulated bidirectional communication between the central nervous system and the peripheral blood supply.
So they talk to each other.
Yes. And when the PLX5622 drug depletes the microglia, the resulting chemical vacuum basically signals the peripheral immune system to mobilize.
The body sends in the National Guard.
Precisely. Studies tracking peripheral immune markers show a massive shift. You see a rapid influx of neutrophils, which are the body's fast-acting, highly aggressive early responder white blood cells, physically breaching the blood-brain barrier and entering the brain parenchyma.
Wow. They just storm the gates.
Furthermore, in the bloodstream itself, you observe a significant shift in monocyte populations toward a highly inflammatory state. The systemic immune system is ramping up, preparing to storm the brain to cover the massive vulnerability left by the missing microglia.
Which proves without a doubt how vital the microglia are to maintaining the peaceful homeostasis of the brain. The moment they are gone, the surrounding cells panic and the peripheral immune system prepares for war.
It really does.
But here is the magnificent twist, the absolute core of the deplete and repeat methodology. Eradicating the bad paranoid microglia is only step one. The true medical miracle is what happens when you turn off the drug and let the brain rebuild.
This is where the therapeutic potential of this research transitions from just being interesting to being completely paradigm-altering. Because the depletion is achieved using a pharmacological inhibitor in the diet, the entire process is 100% reversible.
You just stop feeding them the medicated chow.
Exactly. Once researchers remove the PLX5622 chow and return the mice to a standard diet, the CSF1R receptors are unblocked, and the biological response is explosive.
How explosive?
The microglial population does not just slowly recover over months. It repopulates with astonishing aggressive speed. Within 14 to 21 days, the brain has regenerated a completely full, brand new population of microglia, entirely filling the physical space that was emptied.
Okay, this is the part of the mechanism I desperately need you to explain because my biological math just isn't adding up here.
Okay, what's missing?
Early in this deep dive, you explicitly stated that microglia originate in the embryonic yolk sac, migrate into the fetal brain, and then the gates close forever.
Yes, I did.
So if an adult mouse or an adult human no longer possesses an embryonic yolk sac, where are these millions of new cells physically coming from in just 2 weeks?
That is the exact question that divided neuroimmunologists when these repopulation models were first discovered. If the original factory is gone, how do you build a new fleet?
Did they come from the bone marrow? Did they come from hidden stem cells?
There were several competing theories. A prominent one was exactly what you'd think, that peripheral macrophages slip through the blood-brain barrier and just change their uniforms to look like microglia.
Right. Did they just put on different hats?
Exactly. But sophisticated genetic lineage tracing studies where researchers essentially tag the DNA of cells with glowing fluorescent markers ruled that out completely.
So it's not the peripheral immune cells.
No, the peripheral macrophages that rush the brain do not become the new microglia. The truth is much more fascinating, and it relies on a biological phenomenon called massive clonal expansion.
Clonal expansion. Explain how that works in this context.
Well, remember when I said the PLX 5622 drug depletes 90 to maybe 95% of the microglia?
Yeah.
It never achieves 100% eradication. There is always a tiny, deeply hidden fraction of surviving microglia, perhaps around 1% of the original population.
So there is a 1% remnant clinging to life.
Yes. Some of these survivors are located in specific niches, and some researchers believe they possess a slightly more primitive, progenitor-like genetic signature, expressing markers like nestin.
Okay. What happens when the drug goes away?
When the inhibitory drug is removed, that 1% remnant senses the massive, empty biological real estate surrounding them. The absence of neighboring cells triggers highly aggressive proliferation signaling pathways.
They realize they have the whole place to themselves.
Exactly. These surviving cells begin to divide furiously. They replicate their DNA and divide over and over, undergoing exponential clonal expansion. Because microglia are inherently highly modal, as the clones multiply, they rapidly migrate outward, evenly redistributing themselves throughout the brain. This continues until the tissue reaches its optimal cellular density, at which point a biological braking mechanism halts the division.
So a microscopic handful of survivors essentially repopulates the entire continent in 2 weeks. It is literally a biological reboot. It's the ultimate IT support solution applied to neurobiology. Have you tried turning your brain's immune system off and on again?
I love that. The controlled demolition and rebuild analogy captures the sheer scale of the turnover perfectly.
But the critical question is the quality of the new cells. When this massive new population finishes proliferating and sets up shop, do they remember the trauma? Do they remember the concussion, the torn axons, the excitotoxicity?
This is the multi-million-dollar revelation that makes this therapy viable. No, they do not remember. The rapid force turnover effectively strips away the disease pathology.
Wait,
Yes, it resets the cells to their default factory settings. The brand new microglia are functionally naive. They are completely peaceful, occupying that delicate, highly branched, ramified state.
so they're back to being octopuses.
Exactly. They do not carry the altered epigenetic signatures or the paranoid transcriptional memory of the original traumatic brain injury.
The tripwire is entirely dismantled. The paranoia is gone.
Completely gone. And the cascading physiological benefits of this cellular factory reset are sweeping and profound.
Let's talk about those benefits.
First and foremost, the chronic neuroinflammation abruptly ceases. The elevated inflammatory gene expression that usually plagues a brain for months after a TBI is drastically reversed. The new microglia are not churning out toxic reactive oxygen species. They are peacefully surveying the tissue.
The fire's out.
The fire is out.
And what about the collateral damage the fire was causing? We talked earlier about how the secondary injury causes the mitochondria, the cell's power plants, to fail.
The microglial reboot remarkably rescues the brain's energy metabolism. We can actually measure mitochondrial health by looking at specific metabolic byproducts, primarily the lactate to pyruvate ratio.
Let's define that for the listener, because it sounds like a high school chemistry nightmare. What is pyruvate and why does it turn into lactate?
Pyruvate is a crucial molecule. In a healthy cell with plenty of oxygen, pyruvate enters the mitochondria and fuels the Krebs cycle, producing massive amounts of clean energy, ATP.
Okay.
But if the cell is under severe oxidative stress and the mitochondria are damaged, which happens during chronic TBI inflammation, the cell cannot use oxygen efficiently. It has to rely on anaerobic respiration, essentially fermenting the pyruvate into lactate just to survive.
It's exactly like what happens in your leg muscles when you sprint too hard,
Yeah.
right? You run out of oxygen, your muscles produce lactic acid, and they cramp up.
Precisely. A TBI causes the brain to essentially cramp. The tissue suffers a dangerous toxic buildup of lactate because a key enzyme, pyruvate dehydrogenase, gets suppressed by the inflammatory environment.
And what did the Deplete and Repeat protocol do to this?
When researchers subjected injured mice to the protocol, the new healthy microglia stopped producing the oxidative stress. The mitochondrial function was protected, the enzyme levels normalized, and the toxic lactate buildup was completely prevented. The brain's engine could breathe normally again.
The cellular chemistry is actually repairing itself. The mitochondria are firing, the inflammation is gone.
Yeah.
But all of this microscopic cellular data, the gene expression, the clonal expansion, the lactate ratios, that is all happening invisibly.
True.
Does this biological reset actually translate to the physical lived experience of the animal? If you reboot the immune system of a brain-damaged mouse, do you actually cure the physical symptoms of the trauma?
The behavioral rescue observed in these studies is arguably the most staggering data in the entire review. The researchers didn't just look at cells in a petri dish. They rigorously tested the cognitive and emotional function of living animals.
Okay, let's look at cognitive memory rescue first.
Right. Mice subjected to a severe TBI routinely fail standardized spatial working memory tests. Researchers use a tool called a Y maze to evaluate this.
A maze shaped like a Y. How does it test working memory?
Mice are naturally highly curious exploratory animals. In a Y maze, a healthy mouse will explore one arm, then the next, instinctively remembering which arm it has already visited to maximize its exploration of new territory.
They want to see it all.
Exactly. This is called spontaneous alternation. It requires intact, short-term working memory to map the immediate environment.
And a mouse with a TBI.
A mouse suffering from chronic TBI-induced neuroinflammation exhibits severe deficits in working memory. It will wander aimlessly, repeatedly returning to the same arm it just left, unable to retain the short-term spatial map.
Oh, that's sad.
It is. However, post-TBI mice that underwent the microglial Control-Alt-Delete protocol performed significantly better. They showed rescued alternation rates nearly indistinguishable from healthy, uninjured control mice.
Their short-term memories restored, just like that. What about long-term complex learning?
For long-term spatial reference memory, researchers use the Morris water maze.
I have read about this one. It sounds incredibly stressful for the mouse. You place the rodent into a small circular pool filled with opaque water.
Yes, the water is usually made opaque with a non-toxic white dye, so the mouse cannot see beneath the surface. And hidden just a millimeter below the water is a small resting platform.
And mice hate swimming.
They do not like being in the water, so it swims around frantically until it accidentally bumps into the hidden platform and climbs onto it to rest.
How does that test memory?
Over a series of days, the researchers place the mouse back in the pool from different starting points. A healthy mouse with an intact hippocampus will quickly learn the visual cues on the walls of the room, a poster here, a door there, and use those cues to triangulate the exact location of the invisible platform.
Oh, so they build an internal GPS map of the room.
Exactly. After a few trials, they swim in a perfectly straight, efficient line directly to the hidden safety zone.
And the TBI mice.
A mouse suffering chronic TBI effects cannot build or recall that map. Even after days of training, they swim in erratic, inefficient loops around the edge of the pool, struggling to remember where the safety platform is located.
And the ones with the reset microglia.
Remarkably, the mice that had their primed microglia depleted and repopulated used highly efficient search strategies. Their spatial learning pathways were repaired.
That is just phenomenal.
They also excelled in non-spatial memory tasks like the novel object recognition test.
How does that one work?
You place a mouse in a box with two identical objects, let them explore, then remove them. Later, you put them back in, but replace one object with something entirely new.
And the healthy mouse checks out the new thing.
Yes, a healthy mouse will spend vastly more time sniffing and whisking the novel object because it remembers the familiar one. TBI mice treat both objects as new because their recognition memory is degraded.
But the repopulated ones?
The repopulated mice showed a fully rescued preference for the novel object. The cognitive hardware was functionally repaired.
That is awe-inspiring. Their working memory, their spatial navigation, and their object recognition are all physically restored because the hypervigilant immune cells that were eating their synaptic connections have been replaced with peaceful cells.
Yes.
But what about the emotional toll?
Mhm.
We discussed at length how female mice, in particular, demonstrated delayed, profound psychiatric symptoms like depression weeks after an injury.
The microglial reboot rescued the psychiatric pathology just as effectively as the cognitive pathology.
Using that tail suspension test.
Yes, using the tail suspension test we detailed earlier, researchers measured the baseline resilience of the animals. While the standard post-TBI mice exhibited severe behavioral despair, giving up and hanging immobile,
Yeah.
the mice that experienced the forced turnover of microglia demonstrated significantly attenuated depressive-like behaviors. They regained their innate drive to struggle and survive. The chemical imbalance driving the depression was actually corrected by the introduction of the naive immune cells.
I want to take a moment to pull all of these threads together because the implications of this research are genuinely paradigm shifting for anyone listening who has dealt with head trauma.
It really changes how we view the injury.
Yeah, we are taught to view a traumatic brain injury as a static physical scar. You broke your arm, it healed with a bump. You hit your head, you have permanent brain damage. But this research proves that the chronic, debilitating symptoms of a concussion, the relentless brain fog, the crushing fatigue, the sudden onset of depression, the inability to remember where you put your keys. These aren't necessarily permanent structural voids in your brain.
Right.
They aren't unchangeable scars.
That is the pivotal conceptual leap. They are the downstream results of an ongoing active biological process.
It is an active process driven by confused, deeply traumatized immune cells. And the absolute beauty of an active biological process is that it is fundamentally vulnerable to intervention.
Exactly. It can be stopped.
It can be stopped and it can be reversed. By viewing a brain injury not as a moment locked in time, but as an evolving cellular environment, the prognosis transforms completely. We move from the depressing reality of learning to manage your permanent deficits to the revolutionary possibility of resetting the cellular environment and allowing the brain to heal itself.
It is a profoundly hopeful synthesis of the data, however.
Here comes the reality check.
Yes, as scientists, we have an obligation to provide a stark reality check regarding clinical translation. What we have spent the last hour discussing is an incredibly powerful preclinical research tool.
Preclinical, meaning not ready for humans.
Right. It has brilliantly illuminated the hidden pathophysiology of traumatic brain injury in rodent models, but you cannot simply walk into your local pharmacy tomorrow, ask for a CSF1R inhibitor, and undergo a microglial reboot.
Sadly, the Control-Alt-Delete pill is not yet available over the counter next to the ibuprofen.
And for very good reason. Attempting to execute this pharmacological depletion protocol on a human patient right now would carry astronomical, potentially fatal risks.
Why? Because of the infections.
As we discussed when you brought up the ICU scenario, removing 90% of the brain's innate immune system causes massive systemic shockwaves. But also, we still have a very poor understanding of the long-term effects of systemic CSF1R inhibition on human hematopoiesis.
Hematopoiesis. That's the complex process of creating new blood cells in your bone marrow, right?
Exactly, because the drug wouldn't just hit the brain, it would circulate through the whole body.
It would go everywhere.
Exactly. It would heavily impact macrophage populations in your liver, your lungs, your spleen. You would be inducing a state of severe, widespread immune compromise.
Basically wiping out the whole body's defense.
You would be leaving a human patient highly susceptible to opportunistic systemic infections that a healthy immune system would normally brush off. There is a vast mountain of pharmacological optimization, targeted delivery research, and rigorous safety profiling to be completed before anything resembling this protocol reaches a human clinical trial for central nervous system trauma.
It is the ultimate scientific double-edged sword. We have finally located the kill switch for chronic neuroinflammation, but we still have to figure out how to press that switch without simultaneously turning off the patient's biological life support.
That's the challenge for the next decade of pharmacology.
But even acknowledging the massive hurdles of clinical translation, simply knowing that this physiological reset switch actually exists, It leaves you with a truly mind-bending concept to ponder.
It opens doors to therapeutic avenues we quite literally could not have dreamed of 20 years ago.
Because we have seen compelling evidence today that forcing the brain's immune system to turn off and turn back on can cure the lingering toxic trauma of a physical brain injury. But the source material leaves us with a lingering provocative thought for the listener.
And this is the part that really keeps researchers awake at night.
Yeah. We know that microglia do not just become primed and paranoid from a physical concussion. The literature notes that microglia naturally become primed and hypervigilant simply as a function of chronological aging. They become primed by enduring severe, chronic psychological stress over decades. They become aggressively primed by the onset of neurodegenerative nightmares like Alzheimer's disease and Parkinson's.
That is undeniably true. The exact same hyperreactive, paranoid, synapse-eating microglial state that we see following a TBI is recognized as a central driving hallmark of the aging brain and severe cognitive decline.
So let's extrapolate. If pharmacologists eventually perfect this Control-Alt-Delete drug for human biology, if they make it highly targeted, incredibly safe, and capable of being administered without compromising the peripheral immune system, could we one day cure the natural cognitive decline of the aging brain simply by giving it a brand new, factory reset immune system?
It's an incredible thought.
If we can wipe away the cellular paranoia that causes depression and memory loss after a car crash, what else could we cure if we just learned how to safely reboot ourselves?
If we can successfully domesticate this biological mechanism, the potential to rewind the clock on neurological aging is theoretically immense.
At the start of this deep dive, we talked about how a broken bone is simple. You get a clean x-ray, you see the physical break, you set it, and you fix it. A head injury, however, drops you into terrifying diagnostic muddy waters. The damage is invisible, hiding in the microscopic paranoid behavior of cells you never knew you had.
They're a hidden enemy.
But maybe, just maybe, by learning how to drain that toxic water entirely, scrubbing the pool and letting it refill with pristine new cells, we can finally extinguish that slow-burning fire in the mind once and for all. Heliox is produced by Michelle Brucker and Scott Bleakley. It features reviews of emerging research and ideas from leading thinkers, curated under their creative direction with AI assistance for voice, imagery, and composition. Systemic voices and illustrative images of people are representative tools, not depictions of specific individuals. Thanks for listening today. 4 recurring narratives underlie every episode: boundary dissolution, adaptive complexity, embodied knowledge, and quantum-like uncertainty. These aren't just philosophical musings, but frameworks for understanding our modern world. We hope you continue exploring our other episodes, responding to the content, and checking out our related articles at helioxpodcast.substack.com.